This is Chapter 6, Energy and Metabolism. In this chapter, we will be focusing on enzymes. Enzymes help the cell obtain and use energy and control an organism's metabolism. Metabolism is all the reactions that occur in a living organism, the reactions within the cell and the reactions within the body as a whole. As we've talked about before, all energy within a living system begins with the sun. That energy is converted into glucose through photosynthesis and then converted into other stored energy once that plant life has been consumed. How quickly or how slowly that stored energy is turned into usable energy is determined by enzymes. Before we talk about enzymes, we need to know a little bit more about energy, or thermodynamics. Thermodynamics is the branch of chemistry concerned with energy changes. The first two laws are here on the screen. The first law basically states that the total amount of energy in the universe remains constant. It just changes from one form to another. This is true in living systems as we change glucose to ATP and then ATP into some sort of cellular process. The second law talks about what happens to that energy as it changes. As with all things, energy flows from higher to lower. In this case, if it has a higher energy level, it is less stable. As it flows to a lower energy form, it becomes more stable. During each conversion, some energy is lost as waste heat. As the energy is passed, more and more energy is lost. Eventually, so much energy is lost as waste heat that the energy that remains is no longer able to do work. The second law of thermodynamics includes the idea of entropy. Entropy is the measure of disorder in a system. Disorder is more likely to happen than order because disorder doesn't require any energy, where being organized does. If you look at the picture, you can see which bedroom required energy to keep it ordered and which one had entropy occurring spontaneously. Entropy happens in living things as well. Each time a chemical process happens, waste heat is created. Each time an organelle does its job, it becomes a little more worn down. Over time, if nothing is done, the cell will stop functioning because entropy will have increased to the point where it can no longer operate like it's supposed to. Living things avoid this complete entropy because energy is constantly being resupplied to keep that cell organized. Energy within the cell is created and released through the ATP-ADP cycle. Adenine triphosphate, ATP, is the cellular energy molecule. ADP, adenine diphosphate, is the molecule that's created when that energy is used. So what we're doing is we're adding a phosphate, taking a phosphate off when we create energy and when we use energy. There are two types of energy-related reactions. An endergonic reaction requires the input of energy for the reaction to happen. This is not the same as an endothermic reaction because endothermic reactions involve heat. In an endergonic reaction, the final product has more energy than the starting substances. An example of this is photosynthesis. In an exergonic reaction, energy is released. Again, this is not the same as exothermic because exothermic involves the release of heat. The final products have less energy than the starting substance. Respiration is an exergonic reaction. Cells earn or create ATP in exergonic reactions and spend or use ATP in endergonic reactions. The picture below shows that ATP ADP cycle I mentioned earlier. When the cell breaks ATP down into ADP, energy is released. This energy is used to power the endergonic reactions like muscle contractions in the body. That's why it's called spending ATP. Most reactions require an input of energy to happen. This is because you have to break the bonds that already exist between the atoms in order to allow new bonds to form. This energy is called activation energy. Lowering the activation energy needed can be done in a variety of different ways, including heating the reactants, or in this case, using a catalyst. If you look at the picture, we're turning frogs into princes. The one on the left has a very high barrier. This means it takes a lot of activation energy to turn each frog into the prince. If you add an enzyme to catalyze this reaction, it will bring the activation energy down, in this case bringing the wall down, so more frogs can be turned into princes. Catalysts make a reaction happen faster because they lower activation energy. They don't make any more product than normally would be made, they just make that happen faster. Catalysts also don't get used up themselves, so a catalyst can be used again and again and again. Enzymes are the catalysts in biological reactions. If you look at the screen, the red line is the activation energy of a catalyzed substance. If you look at the blue line, that's the uncatalyzed line. So you can see that by the addition of an enzyme, that activation energy decreases and more product is made more quickly. An organism's metabolism is based on energy. It's the total of all the chemical reactions that are carried out by the organism, and this is a balance between anabolic and catabolic reactions. Anabolic is going to use energy to build molecules, so that makes them endergonic reactions, and catabolic collect energy by breaking molecules down, which make them exergonic reactions. Enough energy must be taken in to be able to build the molecules necessary for day-to-day existence, as well as to store it for future needs. Enzymes help cells to achieve that balance by reducing activation energy. This means that less energy is needed and more energy is left over for storage. These are 4 characteristics that apply to all enzymes. You need to keep these in mind as we talk about how enzymes function. The first thing you need to keep in mind is that enzymes are 3-dimensional. They have pockets on their surface where the substrates bond to them. These pockets are called the active sites. When the substrate bonds to the enzyme, it forms what is called the enzyme-substrate complex. The fit between the substrate and the active site is exact. It's perfectly fitted around the substrate. This increases the enzyme's efficiency. The enzyme-substrate complex allows the enzyme to alter activation energy in 2 ways. First, it can change the orientation of substrates, putting them in a specific direction that will increase the opportunity for them to bond together, which will increase the reaction when you're creating something new. Or they can provide a stress between the chemical bonds that already exist in the compound that's in the active site and actually break it apart. Again, this lowers the activation energy, and so the reaction happens more quickly. This is an exploded version of how an enzyme works. So we're going to start with a substrate, which is sucrose. It's a disaccharide. Remember, we talked about those in Chapter 3. It is glucose and fructose bonded together. The substrate bonds to the active site, creating the enzyme-substrate complex. The enzyme puts stress on the bond between the glucose and the fructose, basically stretching it, pushing it, pulling it until that bond breaks and the individual molecules of glucose and fructose are released. That opens up the enzyme's active site and it's able to repeat the cycle. One thing you need to keep in mind is that enzymes are flexible. When a substrate binds to it, the enzyme adjusts its shape slightly, making the fit between the two even better. This is called an induced fit. The induced fit enhances the enzyme's ability and the reaction moves even more quickly. If you're looking to increase reaction rate with or without an enzyme, you can try changing temperature. If you increase the temperature of a reaction without a catalyst, without an enzyme, the reaction rate will increase because the additional heat increases the molecular motion of the atoms. As motion increases, the atoms will bump together more often and the reaction will increase. If you add an enzyme, that reaction will also increase. It will make the enzymes move more quickly and bond more quickly. But this only works to a particular point. That point is called the optimum temperature. If you are below optimum temperature, the forces that give the enzyme its shape will make it too inflexible and it won't be able to obtain the induced fit. Remember, an improper fit between the substrate and the enzyme will slow the enzyme down and it won't be able to do its job more efficiently. If you are above optimum temperature, the enzyme can't maintain its shape, and so again it becomes less efficient. Another way to influence enzyme activity is by altering pH. pH is a measure of the concentration of hydrogen ions in a solution. Enzymes are particularly sensitive to the concentration of hydrogen ions in the fluid that they are found in, because changing the hydrogen concentration changes the balance between the positive and negative ions within the enzyme itself. Basically what that means is that if you have too many hydrogen ions, they begin to imbalance the enzyme and it begins to come apart. Most enzymes have an optimum pH, just like they have an optimum temperature. And if you are outside of that optimum area, the enzyme is beginning to come apart and it's unable to do its job efficiently. Sometimes a cell needs to regulate the activity of an enzyme, meaning sometimes a cell wants an enzyme working at full speed, sometimes at a slower rate. A substance that binds to an enzyme and decreases its activity is called an inhibitor. Enzyme inhibitors occur in two ways. There are competitive inhibitors and non-competitive inhibitors. Competitive inhibitors compete with the substrate for the same active site. They will occupy the active site so that the substrate can't, preventing the substrate from binding, which makes the entire process slow down. Non-competitive inhibitors will bind to the enzyme in a location other than the active site. This forces the enzyme's shape to change. So when the substrate goes into the active site, it doesn't fit properly, and the induced fit is incorrect. This again slows the entire process down. Enzymes come in two separate categories, those that are on all the time and those that have an on-off mode. Those that have an on-off mode or an active and inactive form are called allosteric enzymes. These work by having a non-competitive inhibitor bind to a specific site on the enzyme. This site is called the allosteric site. When the non-competitive inhibitor is bound to the enzyme, we have an inactive enzyme. When the non-competitive inhibitor is gone, then we have an active enzyme. A non-competitive inhibitor that binds to the allosteric site and reduces the enzyme's activity is called an allosteric inhibitor. It works in exactly the same way as a regular inhibitor, except that instead of slowing the activity down, it turns it completely off. Allosteric activators work in reverse. They bind to the allosteric site and increase the enzyme's activity. Enzymes are cofactors that help enzymes do their jobs. They can be metal ions like zinc and iron. These elements will make the bonds of the substrate less stable so that the enzyme is able to break them apart more easily. They are often found in the active site where they can provide the most useful energy. Cofactors can also be coenzymes. Coenzymes are also found in the active site. They make the induced fit better, and they also pass electrons from the enzyme to the substrate. That movement of the electron also helps the substrate to break apart more quickly. Coenzymes such as NAD, NADP, and FAD are used to help move hydrogen atoms between enzymes as a way to move energy within the cell. For a biochemical pathway to operate efficiently, its activity must be regulated by the cell. It would be a waste of energy and raw materials to make compounds if the cell already has enough of them. It's to the cell's advantage to be able to shut those pathways down when they aren't needed, and that is feedback inhibition. In a normal pathway, the first enzyme alters the substrate, changing its shape. And as we said before, each intermediate substrate along the way changes that initial substrate until we get to the end product. With no inhibition, the final product is continually made. It does not stop. In a pathway with feedback inhibition, when a certain amount of final product is made, it acts as an allosteric inhibitor. It changes the first enzyme shape so it can't make that first intermediate substrate. This stops the entire process. When the final product has been used up, that molecule that's acting as an inhibitor breaks off from the enzyme, is itself used up, and then the process is able to begin again.